Hi, I'm Dr. Elana Miller. Today we are doing a deep dive into the National Institute on Aging's findings on Acarbose and Rapamycin's potential to extend lifespan. There are a lot of interesting findings on which drugs increased lifespan and which ones failed that I'm excited to share with you. Grab your lab coats and let's get ready to dive into the world of longevity molecules. The National Institute on Aging's Intervention Testing Program, or the ITP, is the most exhaustive testing framework and system to evaluate whether certain molecules can extend lifespan in mice and to understand the underlying mechanisms leading to those benefits. The ITP evaluated a selection of promising longevity molecules, including rapamycin, metformin, nicotinamide riboside, and the SGLT2 inhibitor canagliflozin, among others. And you know what? Two of those molecules, rapamycin and acarbose, showed a significant increase in lifespan in the mouse model they were studying. But here's the thing. Metformin, a medication commonly used to maintain blood glucose levels, failed to show positive results on lifespan. So why the difference? Well, it turns out that there's something about the exact way that acarbose lowers glucose levels through its effects on gut microbiota, which makes it a powerful health span promoting molecule. Let's start by reviewing how acarbose works. Mechanism number 1, regulating glucose spikes. Acarbose works by blocking an enzyme called alpha-glucosidase, which helps digest and absorb carbohydrates in the small intestine. By inhibiting this enzyme, acarbose slows down the digestion and absorption of carbohydrates, which helps regulate blood sugar levels and prevents spikes in blood glucose. Why is this important? As we age, our bodies become less able to regulate glucose and insulin levels. High levels of glucose in the body can increase the risk of cardiovascular and metabolic problems and can also damage the endothelial cells that line our blood vessels. Elevated glucose levels increases the production of mitochondrial reactive oxygen species, or ROS. These molecules, which contain oxygen and can either be free radicals or non-radicals, are highly reactive and can cause damage to cells and tissues. By lowering postprandial spikes in glucose, acarbose reduces the amount of ROS activity, lowering endothelial damage and the overall risk of cardiovascular damage. Mechanism number 2, increasing insulin sensitivity and decreasing IGF-1. By dampening the postprandial peaks in glucose, acarbose also indirectly increases insulin sensitivity. Lower concentrations of glucose means that less insulin is required to transport that glucose into the cells for energy. Elevation in insulin corresponds to an elevation of a growth hormone called insulin-like growth factor, or IGF-1. Both insulin and IGF-1 play an important role in promoting cellular growth. IGF-1 is used by bodybuilders for its anabolic effects, which can help increase muscle tissue. However, it is known that bodybuilders have higher rates of early onset of age-related chronic diseases, such as cancer and cardiovascular disease. Excessive growth signals can lead to the proliferation of unhealthy cells, including senescent and cancerous cells, in addition to healthy cells. Through the understanding of Mikhail Blagosklony's hyperfunctionality theory of aging, we know that most disease states occur when cells grow excessively, overexpress and secrete certain molecules and proteins, and stimulate the growth and replication of adjacent cells. Excessive growth signals from elevated glucose, causing increasing amounts of insulin needed to metabolize that glucose, lead to elevated mTOR activity. Elevated mTOR activation is a feature of aging tissues and cells. When cells are exposed to certain stimuli, such as oxidative stress or DNA damage, mTOR can become hyperactive and stimulate cell growth in an uncontrolled manner. Acarbose dampens the growth stimuli IGF-1, insulin, and glucose. In doing so, it decelerates mTOR-driven aging and the growth of dysfunctional tissue. Mechanism number 3, microbiome remodeling and optimization. There is a growing body of evidence suggesting that imbalances in the microbiome, known as dysbiosis, drive aging. The diversity of the gut microbiota tends to decrease with age, which contributes to dysbiosis. Dysbiosis can trigger low-grade inflammation and has been linked to the development of certain diseases, including cancer. Acarbose increases the amount of resistant starch that cannot be broken down by intestinal enzymes, Resistant starch enters the colon, where it is fermented by the gut microbiota. The fermentation leads to an increase in the production of beneficial short-chain fatty acids and increases the abundance of certain types of beneficial bacteria in the gut, such as bifidobacterium and lactobacillus. Short-chain fatty acids may play a critical role in extending longevity and may also produce other signaling molecules that are associated with longevity benefits. In sum, Acarbose reduces the expression of inflammatory factors by increasing the abundance of beneficial bacteria. The downstream effects on inflammation reduction have a significant implication for longevity. Lastly, mechanism 4, GLP-1 derived cardiovascular health benefits. Acarbose also increases concentrations of circulating glucagon-like peptide 1 or GLP-1. Acarbose also increases concentrations of circulating glucagon-like peptide 1 or GLP-1. GLP-1 is a hormone that is produced in the gut in response to food intake. It improves insulin sensitivity and reduces blood sugar. GLP-1 also seems to act directly on the vasculature, liver, myocardium, B cells, and brain to safeguard the structural and functional integrity of these organs in a way that is likely to slow aging and promote longevity. Okay, wrapping things up. Mechanistically, we now understand multiple pathways on which Acarbose seems to be working, which comports with our understanding of aging as dysfunction in multiple interconnected biological pathways. Rapamycin and Acarbose promote health span and lifespan by targeting aging pathways that are partially distinct from each other. Therefore, these two molecules complement each other well. In 3 independent trials, the combination of rapamycin and Acarbose extended lifespan by 30% in the mouse model. There's more to study, but the research on Acarbose and Rapamycin is becoming stronger and stronger every day. The findings so far are promising and suggest that these molecules may be powerful tools to promote health span. The research is ongoing and exciting. We look forward to sharing more information with you as things unfold, so stay tuned.